Hello and welcome to the first in a series of talks that I'm giving. This one is on microbial biogeography. Start out with a quick karakia. Hei wai whakaro, kia tika, kia ngākou pai. Humie, tai kie. So this is also the Scout law, and it's how I like to set the tone whenever I'm giving a talk, which is have respect, do what's right, and be positive. Let us all come together and move forward. So, over the next series of four sessions, I'm going to talk about microbial biogeography, adaptations extremophiles have to different environments, and how they can teach you how microorganisms adapt and are shaped by their local environment niche selection. This first talk gives you a theoretical understanding of microbial biogeography. Now, of course, we haven't, since we can't see microorganisms with our own eyes, that makes it a little bit harder for us to observe their distribution. So it's only with relatively recent technologies that we've been able to do a lot of this sort of work. So I always feel like we're playing very much catch up. Now, I'll also point out, so I'm recording this in my house on the weekend because the recording didn't work when I was at Massey. Massey doesn't seem to like my computer for one, and just generally wasn't, the system wasn't working optimally last Monday when we tried to do it. So I'm in a house with five children, actually six if you include my daughter's boyfriend, and Lamb off to the side, and it is the weekend, so they are about. Apologies if there are any disruptions related to that. So a little bit about myself. Name is Steve Archer. I'm a senior scientist and it's team leader for rumen microbiology. So that's what I do now, and I joined the rumen microbiology team across the road at Grasslands around about 2 years ago. And it's been a fantastic opportunity to look at a extreme and very interesting ecosystem that also has the ability to be hopefully modified to reduce down the environmental harm of livestock production. So that's kind of what motivated me a little bit. The QR code off to the side is my contact details if anyone is interested as we are going through. Unfortunately, you can't ask any questions as we go through, but I will do my best to be responsive to emails during the course of this time. So yes, my background is in extremophile microbiology. So I've previously worked literally on every continent from Antarctica all the way up to the Arctic regions in North America, across deserts around the world on research vessels. And I've had the great privilege of being able to work in these environments and with a lot of different people. This gives me quite a broad perspective of trying to look at life pushed to its absolute limits. Now, how did I get into this at the first point? I was actually round about where you are now, having to decide what do I do next. I was initially going to be a placement student, probably in one of the CRIs, but my career advisor suggested that I work in a research lab for a while. I got a summer studentship at the University of Waikato, and very much the rest is history. I just loved it so much. And when you ask a 20-year-old kid, how would you like to go out to Antarctica? What would you say to that? So yeah, my that badge in the center is Dr. Steve. I'm also a scout leader, and I think that those different icons within that badge are very indicative of all of the things that have been important to me, but you might find out over the next little bit. On that left-hand side are some of my current projects, looking at kikuyu grass in the bottom there, rumen adaptation plantation of kikuyu grass, working on with the wind wanderers over in Taranaki on biosecurity, and there's a heavy education component of that. And then up the top there is collecting samples to look at gain a mechanistic understanding of aerosolization and dispersion from geothermal sources. Also, of course, very important to me is my family, who no doubt you will be hearing more than what I would ideally like, but it is the weekend. And just a fun fact too, that picture of KFC is no accident, obviously. So I love KFC, but I've had it all over the world, from the northernmost KFC in the Arctic in Cambridge Bay, all the way down to the southernmost in South Street in Invercargill, which is the southernmost KFC in the world. And literally the only continent I have left to have it in is Antarctica, and give me 18 months and I'm hoping that is going to be remedied. So that gives you a bit of an overview of all of the work I've done during that period of time. I've also worked on radionuclides in the ocean off Fukushima, microplastics in the water and air, lots of atmospheric chemistry, including pollutants and their effects on growth of marine organisms, aquaculture. I mean, that's the thing that I absolutely love about science, is the fact that you get such wonderful opportunities to do so much all around the world. So this would usually be the point where I would be putting in a Mentimeter in real time about what do you know about microbial biogeography. So I'd just like you to spend a couple of minutes to have a think about what you currently know. And so, as I say, usually that would be the interactive component, but central ideas to consider and that we're going to talk about is the size of microorganisms mean that their distribution and their dispersion globally are not quite as limited as to larger organisms. Their generation time and population size, they can reproduce with a single individual really, really rapidly, as little as 12 minutes, the fastest replication, and population sizes millions upon millions upon millions. So it's very much this numbers game when considering microbial biogeography, is that you have an absolutely unfathomable number of microbes in any one population. There's going to be dominant components, but there's also going to be a rare biosphere. And all it would take is a change of conditions to make one of those organisms at low abundance to have an adaptive edge to the rest of the community, and potentially they are going to come to dominance and fade back. So biogeography is understanding microorganisms in time and space, but it's always important to know it's a dynamic environment, and most environments do change on a fairly frequent basis. So you're gaining a snapshot in time. So this means that when you're talking about microorganisms and how they're distributed, you can draw broad strokes, but what's there at any one point in time is going to be a very dynamic and interesting thing. So, as I say, it's a subset of biogeography, distributions of organisms in time and space. That video I'd highly recommend that you look at if you've got the time. It just shows a marine organism distributing through the world's oceans, how it's replicating time and time again, and gets absolutely everywhere. On the right-hand side, that figure there is for one of my preprints that we haven't quite pushed over the line to a proper publication yet. But that's looking at the back trajectories, where in the past 3 days the microorganisms have transited from all around the globe to the collection points, which is where those sites terminate. And this is color coded using specialists in GIS to be able to mine the atmospheric modeling to look at the environmental conditions that they have transited over the altitude, UV index, temperature, and so on. Now, this is a great example of this project, how my component is microbial ecology, but I also work with atmospheric chemists. I do my own atmospheric modeling to determine where these organisms have transited. But of course, I need their insight to be able to understand how these models run as well. GIS specialists allow me to actually visualize data sets really effectively. And then we've got ecological modelers who take these sets of data, uh, and they're able to combine it to make some predictive indices. So this particular project was my big global project, and there were over 40 researchers who had been recruited, either due to their ability to access certain field sites that I was able to go to. It's very challenging to access sites effectively around the world in some places. All of their expertise was recruited. So it's a highly collaborative project to be able to look at why you see what's in the air at a particular point in time. And actually, in my later series of talks, I'll be talking specifically about atmospheric microbiology. That's kind of my passion and what I'm world renowned for. So yes, the study of microbial biogeography aims to understand the where, why, and what abundance of microorganisms. Now, uh, there are quite a lot of challenges that are slowly being addressed within this field that I've talked about. Spatial scale being one of them. I love this kind of contrast looking at the on these figures. The top one actually shows a soil particle and how even within the single grain of soil, you'll have different distributions of microorganisms across that soil particle as you're uh, uh, driven by the different chemistry and environments that they're allowing, all sort of working together and providing byproducts to one another, forming this absolutely beautiful microscopic ecosystem, all within a grain of sand. Now, when we're looking at soil biology, soil microbiology, we will take maybe a gram sample. We only need half a gram to get more than sufficient DNA. But that's one of those big questions, is how do we actually take that subset, and how representative is that going to be of a field? For instance, if I went out to a field, took one sample, that might represent, let's say, 80% of the microorganisms that I'd find if I collected another 100 samples within 100 square meters of that site. So you're always trying to take that into account, is that it is as representative as practical. And then that bottom figure shows global dust pathways, showing the movement of dust, let's say, to the United States from all the way over in China. And yeah, the Gobi Desert, so Mongolian desert, the Taklamakan desert in China, and of course also the Sahara Desert coming through from Africa. Down the bottom, you also notice that Australia, being a big desert, we actually get an incredible plume of dust. Australia is constantly dumping on us with these desert dusts that do constantly deposit across New Zealand. And so this is the basis of one of my programs on biogeography. So, yeah, so that kind of carries the trying to define the habitat boundaries across different scales. Now, we've also got a problem of trying to estimate the species richness. So now we use genetic techniques, and I believe in this course that you're actually going to define your own microbial community using amplicon sequencing techniques. This was absolutely game changing. I'll talk about that in a little bit more. But before there were these wonderful genetic technologies, there were cultivation dependent techniques. And cultivation is still really important if we are trying to actually define an ecosystem. By cultivation, this is quite challenging because only about 1% of microorganisms are culturable. Tends to be incredibly laborious. I'm the team leader for human microbiology, and um, the vast majority of these organisms take a lot of labour and specialist technique. It's our point of difference and absolutely fantastic skill set to have, just to be able to better understand those deep relationships amongst microorganisms. But molecular genetics, these genetic technologies, are constantly driving forward how much information we can actually define from an ecosystem at any one point in time. So when I started my undergraduate research, I was working on something called gel fingerprinting ELISA. Yes, this is where you still amplified up the sequence of DNA. Then you used enzymes to cut it into pieces, creating a fingerprint, different banding patterns depending on the biodiversity that you'd find in a particular environment. So this could give you a genetic comparison between two locations, but didn't really define what the organisms were. To do that, you could do something called a clone libraries. This allowed you to define a much higher number of species and actually get a sequence that then you could compare to databases. It's still incredibly laborious, but is was constantly driving forward. And then we move all the way through to amplicon sequencing. So now amplicon sequencing is kind of where we're at, and actually we're now using something shotgun metagenomics where we're able to see amplicon sequencing just takes a marker region, as you hopefully have already learned, and allows you to go against databases to look at what the biodiversity is within a particular sample, the microbial composition of the population. Shotgun metagenomics, on the other hand, takes all the DNA sequences, all of it, and you can look at not only what's there, but other genes related to different functional traits. So that's looking DNA based. That's looking at what you'd call the genetic capability. But then if you're actually wanting to look at what's going on within an ecosystem, you need to do something called metatranscriptomics. So that's where you're actually sequencing the messenger RNA within the sample to look at what's being expressed. But regardless, yes, over gel fingerprinting was pretty groundbreaking at the time. Although you already had with clone libraries, that's allowing you to sequence one type of organism at a time. So that's relying on Sanger sequencing, which if you had two different microorganisms, every time there's a variation in the genetic sequence, it would get fuzzy. So you can't really define multiple organisms at a time. Whereas with amplicon sequencing, next generation sequencing, starting with 454, you're able in parallel to sequence thousands, millions of sequences all at the same time due to some very cool technology you should have covered in other areas. So when we talk about microbial biogeography, you can't go past Baz Becking's paraphrase statement: everything is everywhere, but the environment selects. And so this was a hypothesis that because microorganisms are so abundant and small, they're capable of global dispersal via the air. And the only reason you see different biogeography, different community structures within a particular environment is because of this environmental niche selection. You only see marine organisms in a marine environment because only those organisms adapted to that environment are capable of surviving there. However, that doesn't mean that there's not soil organisms about. They're just in that rare biosphere, which hopefully is a concept that you're very well familiar with, this very, very low abundance taxa. And so if you want any elaboration of that, please do let me know. So microorganisms very much are capable of selecting. This was almost a century ago, and I tell you what, I'm writing a paper right now which is highlighting potential genetic traits showing differential dispersion capability and aerosolization capacity as a actual let's say yeah, a evolutionarily selected trait. So this is a theory that my colleagues and I have been kicking around for a while that seems to be growing evidence at a constant rate. Now, of course, actually, it kind of makes sense when you think about a bioaerosol, what's floating around in the air. If you put a piece of bread out, microbes are absolutely everywhere, and you'll find typically the same kind of molds associated with humans that will grow on bread no matter where they are. And they're able to disperse absolutely incredible distances depending on what their lofting conditions were. You typically find somewhere around 10,000 microorganisms per cubic meter. Um, when we're looking, when we're trying to better understand the theory behind microbial biogeography, we look towards macroorganism biogeographic theory. So things that you might be familiar with, distance decay, species area curves, and Rappaport's rules, which you're either covering elsewhere in this paper or you should have at some point, although I might be testing you a bit more on what you learned in first year. Fun fact, by the way, if you do a PhD in the States, they can actually ask you questions related to anything that you've learned over your entire time in university. So you actually get really fundamental questions in the States for your PhD defense. It's not the same here. You tend to just defend on your own work. Still very fun though. Stresses a lot of people out. I had a blast. Um, and then down the bottom, that's a graphic representation of some theories of the microbial recruitment from Antarctica. A being the null, that there is no recruitment, everything remains fixed in place, not very likely. B, that everything is just pretty much derived from Antarctica itself. C, that it's that these ecosystems are recruited from outside of Antarctica and there's not a lot of motion within. And D, that there's a little bit of a mix of everything and the strength depends on the region. This is actually related to one of my first major aerosol paper program, where this was what we were thinking, the hypotheses we were trying to actually look through, and it turned out that it was actually mostly B. It was mostly locally derived, with a little bit of D looped in there as well. So, distance decay relationship for eukaryotes and bacteria. You see that eukaryotes tend to, well, it's a fairly logical thing. The further away 2 points are, the more likely the communities are going to be different. This is true for eukaryotes, but not, and true for bacteria as well, but not quite to the same strength. This does get a little bit complicated as you are looking at different niches and how much you're taking into account the relative abundance of the different organisms. Species area curves, that generally the richness when you're sampling a larger and larger area increase. Imagine you're within a forest. When you sample in a particular plot, you're likely, as you gain more plots, to gain more biodiversity as you're going from plot to plot. It's very true. You'd expect to see more species of trees and different plant species the further out you go. With bacteria, the total community is already diversity, species richness within a community is already quite high to begin with. And so by adding on more sites, as I was talking about that field example, you might only add on a few new species until you start shifting to completely different environments. And even then, within that rare biosphere, if you sequence deeply enough, you're probably going to see much of the same kind of biodiversity within those sites. Rappaport's rule is kind of cool. This is the idea that the environmental range increases the closer to the poles you go, and decreases the closer to the equator you go. And the opposite is true for the species richness. So you get a much lower biodiversity in Antarctica or the Arctic overall. Then you tend to see at the equator, you see a really high richness, but the organisms there are pretty well adapted for that particular area. And so this was actually Gavin Lear is a microbiologist from University of Auckland, and he actually showed that geographic range and genome size of bacteria taxa decline at warmer climates. So you actually had the geographic range decreasing along with the genome size. You'll see also I put in a lot of papers to cite as we're moving along. If you're interested or really trying to go for that high grade, then these are usually quite good and pretty readable papers. All in all, I haven't put on particularly obnoxious papers. So what you found was all of these trends are present, but less pronounced than you see for animals and plants. So you do see these biogeographic patterns, but they're not as pronounced as you see for those microorganisms. Then a set of and these names, Barber and Casameo and Fiera, are pretty big titans to this day in microbiology. Noah Fiera can send out a tweet, and that's how everyone then should be acting from that point onwards. He's an exceptionally brilliant guy with a lot of incredibly big projects on the go at any one time. I'm very fortunate to work with him on a joint project with Colorado State University. Very bright guy. So they published this paper, and this was a great paper from round about when I was doing just at the end of my PhD, where they were looking at how to apply macroecology to microbes for large-scale ecology and biogeography. And this highlights as well some benefits of microbial communities. Because there's larger numbers of species, you can see the niche filtering really happening at a much more dynamic scale. There's huge databases defining microorganisms. So just by that genetic sequence, you can put it against the database, as you'll find, and get an idea of what other environments these organisms have previously been found. And this means it can be really defined, traceable testing of hypotheses with all the standardized data. So looking at, and hopefully if you've got an idea, you've reminded yourself of some of these, this basic ecology, when you're going from a global pool of organisms to a regional pool to a local community, that global pool to a regional pool, so literally global to regional, has got a lot to do with dispersion and speciation. So you have speciation within a regional pool becoming more defined over time, and there's a certain amount of deterministic processes which are filtering for the organisms to get established within a regional pool. They have to at least be partly adapted to that environment for them to actually have any competitive advantage. Once you're going from that regional pool, what's found in a larger area, let's say New Zealand as a whole, to a local community. On top of that abiotic deterministic filtering, you also have biotic niche filtering, so this is competition, all of that. And this is where you get stochastic processes, you get genetic drift, extinction, and dispersion. So this is kind of a classic way of looking at how this ecosystem filtering, how our ecosystem gets structured. It's a combination of historic, deterministic, and stochastic processes. So just have a quick look up and remind yourself of these processes um, if you're still unclear. The neutral model for macroorganisms is that final place where you're looking at from a regional pool to a local pool. You have colonization, extinction, and drift. However, when you're looking at microbial biogeography, the neutral theory would be cutting out that regional pool because dispersion is not so limited. So you're able to go from a global pool distributing potentially very far away all the way to the local community. And so there's some great examples of this. One of my favorite ones actually comes from a study looking at the microorganisms found on top of Mount Erebus, which has got these geothermal fields in Antarctica, not far from Scott Base, isolated by hundreds of kilometers to the next geothermal site. And of course, Antarctica itself is heavily isolated from the rest of the planet by the circumpolar vortex. And yet, what you see up on the top of Mount Erebus is that you see much of the same microorganisms between these global environments and what you actually see on Antarctica itself. So there's a lot more room for sort of random event stochasticity. Um, and the abundance of individual taxa is governed by random speciation events and extinction events, dispersion, and ecological drift. Now, finally, we're looking at this paper by Martini. This was actually one of my favorite papers during my master's. Yeah, so it's just after I did my master's. And so this was looking at is everything everywhere, and what hypotheses should you be thinking about when you're looking at the dispersion of these, first being the null hypothesis. So if we look at that grid down the bottom, and this will come back in later lectures, you have random distribution of microorganisms. So the green is location, the letters are the niche. So when you've got this mix of letters and colours, green to white being different geographic location, let's say Australia to New Zealand, a random distribution looks like these populations are pretty much the same. If there is a very high environmental legacy, so this is highly niche filtering, you'd see that bottom left where the letters cluster together regardless to the location. The legacy three hypotheses, this is looking at historic, what has landed where. So there's a split between Australia and New Zealand, and there is not a continuous flow of organisms between these two environments. So you just have them separated out over a long period of time. And the fourth hypothesis, typically this is how it works, is that there's a combination of 2 and 3. You see that there is some geographic isolation, but then they do still filter into their different niches. So I'll talk about this in a little in a further talk. This is an example of niche filtering at one of my meltwater ponds in Antarctica. And what we saw was when you go down, and this is very, very little pond, 1.3 meters deep, you actually found that heavy geochemical gradients, conductivity, pH, oxygen concentration, heavily filtered these different niches. But then when I compared Huey Pond, which was Bretina Island, to another pond 40 kilometers away, along with similar environmental variables, we found that although the populations were different, they did still kind of fit into the same overall pattern. So there was a heavy amount of niche selection. This was with incredible chemical drivers. So we'll talk about that a little bit more. and future talks. Some more examples of where you're seeing very much niche selection, where there's differences between bare soil, soil crusts, and endoliths. This paper was absolutely fantastic. This looked at a moisture gradient from a lake in Antarctica. And what you're seeing is different niche and neutral theories that are distributing, that are arranging the community structure differently. These are different populations. So we've got actinobacteria, acidobacteria, bacteroidetes, chloroflexi cyanobacteria, firmicutes, and proteobacteria that are all showing different patterns depending on this moisture gradient. So when you're going from left to right, you're going from a low to a high moisture abundance. And you see, for instance, cyanobacteria absolutely love this. They're very niche driven. And then organisms like chloroflexi, they're more like the D. They don't really see much difference in that along that moisture gradient. They're just as adapted as the next one. So what you'd see is A, B, and C being different niche filtering. You have either abundance decreases, increases, or peaks at a particular area along that gradient, whereas D and E are more stochastic. So firmicutes, for instance, kind of has more of that random distribution like E, and chloroflexi has that D. The moisture gradient doesn't actually overall affect that community structure. So it's important to really understand that different populations within a microbial community are going to have different drivers, and different peak niches, and that's how they're going to structure the same bulk community richness. The types of organisms are probably going to remain round about similar, but this niche filtering can act along a gradient, changing the population structure. So quite interesting. So if you were to summarize all of this down to this final slide, hopefully you've gained an idea uh, that some microbial patterns, distributions, and biogeographic patterns, uh, do fit within the macroorganism theories. So you do find a certain amount of logic, although um, not quite as strongly as for macroorganisms. Assumptions are difficult to make, so they've got some compare and contrast the different characteristics of these organisms, microorganisms to macroorganisms, and this just changes the capability to disperse, as well as with a numbers game, the ability to come back from the rare biosphere. Microbes are massively diverse. So they're in a single teaspoon of soil. There is an incredible biological richness, thousands of different species, and trillions of microorganisms in a teaspoon of soil, depending on how rich the soil is. Um, but we have a lot left to understand, but we're moving hopefully in the right direction for all of these patterns as we go through. So thank you very much for your attention. All of my work sort of revolves around an incredible diversity of different people. But I'm very thankful to be able to have this opportunity to share to you. And I hope over the next few sessions, you're going to get a much better grounding of what it's like to be a microbial ecologist, especially in an extreme environment. Please do touch base if you have any other questions. Thank you very much. Take care. Bye.